In this video, we will discuss about one of the approach that has been used to prevent HIV1 infect the host, and that approach uses TRM5. So let us first understand how this TRM5 works to prevent HIV1 from getting infected or from preventing it to infect the host. So there are two types of TRM1. One is TRM5 alpha, another one is TRM-CyP. CyP means cyclophilin. So how this TRM5 alpha works is TRM5 alpha has this V1 loop. There is flexible V1 loop, and here it is the detail of the this V1 loop. It has two part. It has two part. One is SPRY domain, another one is V1 loop. So this is the flexible loop, and that can bind to the uh, N terminal domain of the HIV1 uh, capsid, as it can be shown, as it is shown here. And uh, this is how TRM5 can detect the HIV1 capsid. The second one is the TRIM-CyP, and TRM-CyP has this cyclophilin binding loop, as it is shown here, two cyclophilin binding loop, and it is shown here in the we can say uh, two-dimensional secondary protein structure. This is also a secondary protein structure. Type A is bound with HIV1, CA and terminal domain. CA means capsid protein and terminal domain, and this also interact with capsid protein TRM5 alpha. And this cyclophilin binding domain can interact with capsid protein and terminal domain of the capsid protein, as it is written here and also shown here. And this is how TRM CyP all interact with or can detect the HIV1 capsid protein. And this figure here is shown as how this TRM5 can interact with the HIV1 capsid protein. The green one is the HIV1 capsid protein, and the pink one is the TRM5. So now let us understand this how this TRM5 helps in getting rid of this HIV1 virus. So there are three approaches that are being published, and first one approach is the premature uncoating. So what happens in this case is that nucleocapsid gets unmatured. uh, Nucleocapsid interacts with TRM5 alpha, and that leads to premature uncoding. And what happens because of this premature uncoding is that reverse transcription does not occur properly, and um, and because of this further process, that is host genome integration, does not occur. And this is one of the approach how TRM5 alpha works. Another approach is of the proteasome degradation. And what happens in this case is that this TRM5 alpha helps in attaching the polyubiquitin protein on the capsid protein of the HIV1. And this polyubiquitin then directs it towards the proteasome, and the function of the proteasome is to degrade the protein. So it will degrade this whole capsid protein and its genetic material, that is single stranded RNA. So this is the second approach that has been uh, presented or that has been published. And the third approach is the virography approach, or the virophagy approach, not virography, virophagy approach. And what happens in this case is that the nucleocapsid bind to the isolation membrane. The isolation membrane contains has this LC3 gene. This is the autophagy factor. It will promote autophagy. So this LC3 gene binds to the outer membrane of the HIV1 and also integrates inside the nucleocapsid. And this LC3 gene is the autophagy factor. It will promote the autophagosome. What is this autophagosome means? Autophagosome means that it will prevent the cells it will prevent and make work towards the cell survival by degrading the uh, organelles that are harmful to the cells, organelles or proteins that are harmful to the cells. And one of the approach that it used is autolysosome. So in this case the autolysosome occurs, and because of this LC3 factor now available uh, across this HIV1 nucleocapsid and the outer membrane. So the autophagosome promotes the degradation of the protein that leads to uh, autolysosome or the degradation of the whole virus. So this is the third approach that has been published in the paper. So this is how TRM5 mediated restriction for HIV1. So that's all for this video. Thank you for your time.